The overall goal of the following experiments is to generate RNA substrates for the study of Cas endonucleases, which play crucial roles in the CRISPR-Cas immune systems found in bacteria and archaea. To obtain RNA substrates for Cas endonucleases, PCR amplification of a specific CRISPR region can be employed to obtain a long DNA template for in vitro RNA production. If preferred, 2 oligonucleotides can be annealed to generate shorter templates for substrate RNA production. Alternatively, short, custom-synthesized repeat sequences can then be tested in Cas endonuclease assays. Ultimately, the production of RNA transcripts and their subsequent cleavage by Cas6 endonuclease can be visualized by autoradiography.
The main goal of the presented techniques is to enable researchers to study CRISPR-RNA processing with RNA substrates of varying length. Therefore, different methods are utilized to generate small RNAs, for example single repeat elements, or larger RNAs, such as CRISPR-RNA transcripts that span several spacers.
Though these methods specifically provide insight into CRISPR-RNA maturation, similar template preparation strategies can be adopted for other RNA processing systems.
Demonstrating the procedure will be Judith Zoephel, a graduate student, and André Plagens, a postdoc, both from my laboratory.
In bacteria and archaea CRISPR immune systems, a viral DNA sequence called a protospacer can be inserted between 2 repeat elements in a growing CRISPR cluster in a process called adaptation. Next, the entire CRISPR cluster is transcribed.